Hello learners, in our today's class we are going to learn the technique of RIA, that is radio immunoassay. Before we start on with this technique, we have to revise the concept of competitive ELISA which we learned in the previous video. There are slight of changes in the whole of the protocols of competitive ELISA which makes it much significantly different from radio immunoassay. Earlier we were confused about whether what are the differences which exist between RIA and ELISA, but now after revising the concept of competitive ELISA, you will be very clear with what are and what can be the differences between competitive ELISA and RIA. So I will revise the concept of competitive ELISA here. So there were slight mistakes in the previous video related to competitive ELISA. So let us try to repeat it and revise it. Now what do we do in this process? We have to detect that whether a particular patient has the antigen X or not. And here we can even detect the concentration of that antigen in the competitive ELISA. So we have to first of all detect whether the patient has that X antigen or not. For that, let us label that antigen or let us mark it with pink color. So suppose these are the antigens which are present in the particular serum of the patient. Now what we do is, because we have to detect the presence of this X antigen, we will try to create a specific antibody which is very specific to X. So we will artificially introduce that antibody. This antibody will be artificially introduced into the patient serum. So after extracting the serum, which may or may not contain the X, now what do? We have to take this antibody and then we have to introduce into this tube which contains what? Which contains patient's serum. So suppose now here I have this antibody. What happens is, if this antibody goes inside, it will bind to the antigen if the antigen is present. Now suppose I have introduced a particular amount of antibodies. Let's say I have introduced 5 antibodies. Wait, I have introduced 5 antibodies because it is all the game of concentration. So let us say, on a very hypothetical basis, I have only introduced 5 antibodies inside the serum. Now I had the 5 antibodies. So if the concentration of antibodies is very similar to that of antigen, what happens now? Wait. Let me make this clearly. So see, this was my antibody. Now if the concentration of that particular antibody was very similar to that of antigen, so what could have happened? All the antibodies which we had introduced into the solution will now deliberately bind to the, let me make this a bit smaller. So this particular antibody which was introduced into the solution or which were introduced into the serum now will bind to the what? Will bind to the patient's antigen. So here we have this, then one of them will bind here and then one of them will bind here. So in short, what I want to conclude is that now we will have no free antibodies, no free antibodies available. Why? Because all the antibodies have bound to the particular antigen. Now what do? This has been done. Now we will introduce all of this into the well, where we have what we have done. I'll tell you. So suppose I'm introducing this, then I'm introducing this, then let's say this and then this. So I've emptied it entirely and I've introduced everything here in the well. Now there is one difference you need to observe. In the well, we have already coated the particular antigen which which we wanted to detect. So suppose we wanted to detect the antigen X. So this is the antigen X which has been coated inside the well already, pre-coated in the well. And this is not obtained from the patient, this is obtained from outside. Now what do? We will now take the antigen and antibodies which we had mixed in the serum and we will introduce into this well. Now if the antibodies were already, let's say, bound to the antigen, so what will happen is no of the antibody is free. Means if you can clearly see here that this antibody is bound to this antigen, this antibody is bound to this antigen, so no free antibodies are available. As a result, the antigens which we have coated inside the well will not bind to any such antibody. After this, what do? We introduce the secondary antibody. Now a secondary antibody is the one which will contain the enzyme. So here let's say we have added this as a secondary antibody and it contained an enzyme here. So this is my enzyme and this is my secondary antibody. So I added it. Now what will happen is, as in the concept of indirect ELISA also, the same will be happening here. So let me mark it like this. Wait. Yes. So here it will bind. Similarly, one will bind here, one will bind here, then one will bind here. Means the FAB region of secondary antibody will bind to the FC region of the primary antibody. And now if I add substrate, if I am adding the substrate, let's say, and let's assume the substrate be of light blue color. This is my substrate. Now what I am doing is I am adding this substrate here. So the substrate will be binding to its particular enzyme. So here also it has bound, here also it has bound. Similarly, here also it has bound, here also it has bound, and finally it has also bound here. So in this case you see that all the substrates have bound to the secondary antibody's enzyme which was linked to the secondary antibody. Now what will happen as a result? After all of this, a peculiar color should appear. Let's say the color is of this color. So here the color starts to appear. Suppose this is my appearing color here. This is all appearing inside the well. So this is my color which has appeared. Let me shift it a bit. Okay. So the color has started to appear. Now what will happen? After this, we will do the washing step. As soon as we are doing the washing step, because the antibodies were not bound to the coated antigens, it means all the antibodies along with that color will be washed off. So it will be removed. And what we will observe after the washing, we will observe that there is no color which was produced inside. Getting it? So the color has almost become nil. So I can say that if the color is decreasing or if there is no color, I can say that the concentration of antigen X in the serum, means in the patient, was high, and because the concentration of the antigen in the patient was high, all of that bound to the introduced antibodies. So this is the basic concept of competitive ELISA. Now what we learned yesterday was actually the RIA, that is radio labeled immunoassay. Now what do we do there? Let us come to the concept of RIA. So let me take another page for this. Here we are going to learn the concept of radio labeled immunoassay, that is RIA. So let's learn here what do we mean by this technique. See, basically it is one of the most sensitive techniques which you can perform, because it is very sensitive to that particular amount of antigen or antibody. It can detect very low amount of antigen or the antibody in the patient's serum. About how much? Even lesser than 0.001 microgram per ml. Even lesser than this concentration can be detected by RIA. The RIA was first used by Burson and Rosalind Yellow. They used this and they introduced this technique in 1960, and they were working on the insulin hormone, so they were performing this RIA on that only. So this was done by Burson and Rosalind Yellow, and this was done in 1960. So what do we conclude about RIA here from the two statements? One is that RIA is very sensitive assay, wherein it can detect very less amount of antigens and antibodies also, even lesser than 0.01 microgram per ml. Secondly, RIA was first performed by Burson and Rosalind Yellow. They performed it in 1960 and they wanted to detect the concentration of insulin hormone. Now we talk about the principle of RIA. The principle of RIA will depend upon the competitive binding. So here if I talk about the main principle behind RIA, it is the competitive binding. And competitive binding between whom? So one of them is the normal antigen and one of them is radio labeled antigen, which I can mark it like this. This normal antigen is taken from the serum of the patient and this has been artificially introduced into the particular solution. Now what will happen after this? So let us try to understand the procedure of RIA. It is not that complicated. We will easily understand that how is RIA actually performed and why is RIA performed. So yes, here I am writing the procedure. See, first of all, what happens is that a particular specific antibody, because right now what we need to do is, again we have to, let us say, determine the concentration of one antigen X. Now let this antigen this time be a hormone. So we have to detect the presence of that particular hormone which is acting as the antigen. Now what will happen is, so I can write it as, let's say, H only. H stands for the hormone antigen. So this is my hormone antigen. Now we will take, artificially we will take one antibody which is very specific to that hormone, and we will introduce it into a tube wherein we are going to also add the radio labeled hormone. Now this is radio labeled, it is tagged with some of the heavy isotopes so that it becomes radioactive. So this particular antibody, which is very specific to H, will bind to this radio labeled hormone. And this complex now is very crucial for performing RIA. We ensure that at least 50 to 70% of the radio labeled antigen or the radio labeled hormone molecule must bind to the antibodies which we have provided them. Now what we will do, after this we have to introduce the test serum. So all of this is happening again inside a well. So let us draw here a well. Suppose this is my well, and here I have done what? Here I have added the specific antibodies as well as I have done the hormones and adding of the hormone to that specific antibody, and now I am introducing the normal serum. So let me zoom it into it to explain you in a better way. Okay, so here what we have done is, we have, let's say, the antibody. Let's assume this is my particular antibody. These are my, like, these kind of antibodies I do have. Okay, now they were bound to what? They were bound to the radio labeled hormone. So this is my radio labeled hormone. These were my radio labeled hormones which were comparatively weaker than the normal hormones. Now what we are going to do is, we are going to then bind this and introduce the normal hormone, that is, let us say it is of light blue color. Now this is in the excess here, so this is present in excess, suppose, because we have to detect the concentration. So in the serum, if we assume that this hormone was present, now what will happen is, this hormone will do the competitive binding with this hormone. So this hormone, as a result, this one will be removed, radio label will be removed, and the normal ones will bind. So wait, so this will be again be removed, this will be removed, and then this will be bound to here. Then similarly this one will be removed, this will be bound to here. Then wait, this will be removed, this will be bound here, and similarly this will be removed and this will be bound here. Now what will happen is, after this we can perform various methods wherein we can separate these particular removed radio labeled antigens from the solution, so that only these remain. Now there are many methods, but one of the most popular methods which we perform is solid phase method. Now what is the meaning of solid phase method? Solid phase method is basically a method of separation only. So in solid phase method, what do? We are particularly attaching all of this, the antigen antibody complex, to let's say the plastic wells, these plastic wells only. So what we are doing is, we are trying to coat it inside the well, we are trying to attach it on the well somehow, like this. Or the another alternative which can be done is that we can take the what we call as sapphirose beads. So there are some sapphirose beads, let us try to draw them. Wait, suppose this is my Sephoros bead here. Now what will happen, that this particular will bind here, suppose, then this will be binding again. Okay, wait, let us not make it complicated. Let me delete this Sephoros bead. Let us attach it here only. So this will be attaching here, this will be attaching here, and similarly this will be attaching here. So Sephoros beads can also be added, but we can also either coat them. So solid phase method can either include coating the antigen, means you can coat it in the well, so you can say it as well coating, or you can add the Sephoros beads, Sepharos. Now we will do the washing step. In the solid phase method, now we do what we do washing, wherein this, because it is not attached, it will be removed eventually. So as it is removed, only this part is left, and we say that the radioactivity which was present earlier now decreased and became nil if the concentration of hormone was very high. So now let us plot the graph for the same, that how we can plot the graph here. So if we need to plot the graph, this will be our y-axis, similarly this will be our x-axis, and then we are going to plot it. So suppose we are plotting it with green color. So in downside we'll write the concentration of unlabeled antigen. So here we have the concentration of normal hormone, normal antigen or normal hormone, and here we have the percentage of radio labeled antigen bound, percentage of this kind of antigen bound to whom, bound to the antibodies. So as the concentration of the normal hormone will increase or normal antigen will increase, this percentage of binding will eventually decrease, and this will result into this kind of graph. So this is the graph which we obtain. And why is it happening? Because as the concentration of normal antigen is increasing with time, the percentage of the radiolabeled antigen which is bound is decreasing with the time. So this kind of graph is typically obtained. Now in the book also, here they have given a particular example. So what they have done is they have tried to actually detect the presence of hepatitis B surface antigen. So hepatitis B surface antigen, that is hepatitis B surface antigen, and its concentration in nanogram per ml. They needed to detect whether this is present in the serum or not. So they have plotted the graph of this hepatitis B surface antigen with the hepatitis B surface antigen which is radio labeled. So here we have the percentage bound hepatitis B surface antigen which is radio labeled. So again the similar kind of graph will be obtained. So this is the core principle and the working of RIA. Now if I say that what else can be said here about RIA, so we can also add one more thing, that is that we can talk about the advantages of RIA, or what are the applications of RIA. First of all, the sensitivity of RIA is very high as compared to the other assays. Secondly, it is able to detect the minute concentration. Even the minute concentration of antigen can be easily detected by RIA. And thirdly, it is very highly specific. One last important thing, that is the point of differences between RIA and ELISA, that is RIA and competitive ELISA. So if I write here RIA and if I write here competitive ELISA, this you can fulfill yourself also, but for the sake of simplicity let me mention one of the most important points. In RIA we are using the radio labeled antigen, right? We are doing the radio labeling, so this can be detected only by some specific instruments, for example gamma counter or the beta counter, because these are the radioactive rays, so these cannot be detected normally. And here we have, because we have color here, so it can be detected by the spectrophotometer. Here the radioactivity is not involved. Then remaining is the protocol difference, which you can already write, and also about the specific principal difference, which you can easily now correlate. So this is the main working and principle of RIA, and also that how is it different from ELISA.